Last week, we covered quorum sensing, and on a fundamental level, quorum sensing is just regulations of gene expressions based on population density. So it's how bacteria turn on and off genes depending on how dense they are as a population. So for example, a bacterial population is very, very small. There's just a few hundred cells there. After an extended period of time, if there is sufficient nutrient concentration in the medium that they're in, that population will reach, thousands. And once it reaches a certain threshold number, there's going to be enough of these signaling molecules in the solution surrounding these bacteria to where they'll start enacting their either LuxS, LusR operons. In turn, they will start encoding the genes that are tied to quorum sensing. Those genes could be of multiple different types in nature. So one could be virulence factors, so creating some sort of pathogenic response for bacterial pathogens. Another one could be forming a biofilm, so the bacteria have to communicate with each other in order to work together to form that biofilm. And also, an example of biofilm formation is the plaque on your teeth. So it's very, very common that bacteria form these biofilms. And also, speaking of your teeth, all the different types of bacteria in your mouth, the over 700 different types, they use autoinducers and quorum sensing to communicate with each other in order to, I guess, stage certain things happening within the broader bacterial population. So we're specifically focusing on gram-negative quorum sensing. Gram-negative bacteria use a specific type of quorum sensing molecule called autoinducers. And AI2 or autoinducer 2 is our molecule of choice because it has the ability to transfer across multiple different species, one of which is Salmonella. And this has a certain implication with our project because we're trying to determine if we have a Salmonella infection within our sample of food processing. And I also included a picture of the long LuxR, LuxS operon and AI2 pathway that occurs in E. coli. I don't expect you to remember most of this. If you've forgotten or if you want to learn a little bit more about it, visit last week's video / lecture. I go into it much more detail there. Okay, so last week, I don't know if any of you guys did this, but I asked for example responses. So something the bacteria would do as a response to add to the autoinducer pathway. So something you'd add if the bacterial population gets to a certain size, you want something to happen. What would those things be? If any of you guys did this, please say them now. I'm going to go into the ones I came up with. Okay, I'm gonna go with no one did it. Okay, so the first one I came up with was electronic reporting. So you could program or genetically modify your bacteria in order to output some sort of electrical response. And then you could measure that potential, or you can measure the current in order to determine whether or not you have a sufficient response to act on it. The next one's bioluminescence, which is a little bit cooler than fluorescence. So with fluorescence, you need a UV light in order to see it, or if it's good enough, you'll be able to see it in ambient light. With bioluminescence, like bioluminescent worms or fish or squid, they create their own light. So you put these cultures in the dark, and then you can see them quite well. I will show a picture later. And then finally, what I came up with are converters. So what converters are, they turn a certain chemical into another chemical. Usually you want these chemicals to be, or I guess the produced chemical to be a different color or something that you can measure using other equipment. This is an example of the bioluminescent bacteria. I don't know what type of bacteria it is, but as you can see, it's emitting a blue color. And then this is an example of the converters where you start out with a more clear solution, and as the converter bacteria start transforming that chemical, that clear chemical, further along the line, it starts becoming more and more yellow. So that could be a way you can measure the intensity of that I guess a type of fluorescence in order to measure how successful your transformation was. Okay, I'm going to hit bioremediation now. Bioremediation is, put simply, the process of using microbes to break down pollutants and fix environments by doing so. There are two main different types of bioremediation with microbes. One of which is the native type. This is where you find these native bacteria within their native environment, and you might edit them just a little bit, or you give them the nutrients required in order to be very, very sustainable within their environment. So it gives them sort of a boost in growth in order to help the environment. The second is introduced. So we're now taking these microbes from a different environment, and we're introducing them to an environment that we think that they could help with. Now, of course, within the regular population, right, normal people would be typically okay with native introduction because you're just modifying or helping out the native bacteria. But most people, like with GMOs, are typically against introduction of I guess you could call them almost invasive species. And that's for a good reason, right? Because if these bacteria end up over colonizing their environment that they were introduced to, it could throw off the ecosystem, the soil ecosystem, the water ecosystem of the environment, and in turn have sort of collapse in the biodiverse structures. So just know though, they introduced are still useful. Specifically for stuff that doesn't really matter as much. Say you're working in a plant that wants to use some sort of CO2 scrubbing, you don't really have to worry about an environment there because for the most part, it's a sterile environment. So you're using these CO2 scrubbing bacteria in order to get rid of your CO2 emissions. And this has become, this bioremediation has become even more important over the past few years. Due to a lot of things, but here are a few. One is microplastics, which is actually my favorite. Microplastics are just like sections of water bottles that get broken up due to erosion via water or rocks or sediment and stuff like that. These microplastics contaminate water supplies and they cause reproductive issues in humans along with hormonal imbalances. Just a quick little thing about that is humans intake tons of this stuff. And actually the net increase or intake of microplastics is in plastic water bottles. So every time you drink out of a plastic water bottle, you are getting a huge dose of microplastics. So we're trying to use bacteria to break down these microplastics to save ourselves and also save the aquatic life. Also very, very popular is CO2, which causes global warming. There are bacteria out there that CO2 scrub, so they convert CO2 into a different form or they just consume it. And they use it for cellular respiration. There's also chemical spills. So this can be like an oil spill. Pretty much if you name it, it's probably an issue. Fertilizer runoff, that could be a chemical spill that causes a lot of algal blooms, which in turn cause toxins to fill up the waterways. And finally, heavy metals. So these are metals like mercury, cadmium, arsenic. Those are all heavy metals. And they change the pH of the environment. And for the most part, these metals are toxic to humans and also aquatic life, especially in large quantities. So what happens is so bioaccumulation happens. So these small minnows just get a little bit of it. And then the bigger fish eats tons of small minnows, gets a bigger dose, and it keeps on going up the food chain till you hit like sharks who have tons of mercury in their systems. So we're using bacteria to either fix or do redox reactions with these heavy metals in order to make them more soluble or less mobile. And I included a few pictures. So these are the pictures for the microplastics, smog, so CO2, oil spill, and also this is a fertilizer runoff. So these different bioremediation, how it works, there are multiple different processes. There's not really a set type, but for the most part, bioremediation falls under redox reactions. You should have covered this in chemistry, and pretty much redox reactions are the exchange of electrons to make chemical compounds more stable. And so bacteria can use enzymes in order to make these redox reactions happen faster. It catalyzes them. In turn, we can use these bacteria that have these enzymes to make redox reactions and therefore start helping the environment from pollutants. There's also, I guess this is part of redox reactions, aerobic. So bioremediation processes can be categorized as aerobic and anaerobic. I did not include anaerobic due to slide limitations, but for the most part, aerobic, of course, it uses oxygen. And it specifically, oxygen is used as the electron acceptor in the redox reaction, with the pollutant being oxidized. So the oxygen is being reduced. A few of the different materials you could use during these aerobic bioremediation processes are petroleum, polyaromatic hydrocarbons, which are found in coal. They have a very beautiful structure. And then also phenols, which are just 6-carbon ring with an hydroxyl group attached to it. So finally, there are heavy metals. So this is its own sort of set of bioremediation process. Because heavy metals are a little bit different than these other two. They're much more harder to deal with, actually, due to their heavy nature. It's harder for microbes to bind to them. And our goal with bioremediation with heavy metals is to limit the spread in waterways and also the soil by either reducing the metals through redox reactions, or we can have the bacteria attached to them and then skim that bacteria out of the system in order to remove those heavy metals. It kind of makes them insoluble, so they float to the top or float to the bottom. Float to the bottom. They sink to the bottom. So think of this in this way: is a microbe plus its enzyme is a sort of catalyst to do these redox reactions. So I thought I would include a team that's pretty close to us. It's the University of Nebraska-Lincoln. For the past 3 or 4 years, they have been doing pretty much nothing but bioremediation-ish projects. In 2016, they did a denitrification project of waterways. So nitrification, nitrogen is very, very common in fertilizers because plants need it. And so what happens is this nitrogen runs off when it rains and it goes into the waterways, and it can be very, very harmful for aquatic life. So they're using these bacteria to break down and measure concentrations of nitrate in water. In 2017, they did the reduction of methane. I didn't read too much about that project, but I know that their predominant concern was the methane emitted by cows due to Nebraska having a lot of farmland. And so what it would do is it would either break down or bind to that methane. And finally, in 2018, they did a project very, very similar to us. I think actually we were doing it around the same time in 2018. They wanted to create emerald ash borer traps. And by making traps, I mean they were trying to synthesize the chemicals to make the traps. So these are different types of phenols and stuff that attract these emerald ash borers to the trees. It's what the trees naturally produce. And they're trying to synthesize this substance. So that's kind of a little bit less of a bioremediation project, but you are trying to save the environment from an invasive species. So I guess it falls under an environmental project. If we want to contact them, this is mainly for the Human Relations Committee, but UNL is a great candidate for collaboration. I included their contact information here. We have collaborated with them in the past, specifically when we did the Emerald Ash Borer project. Okay, so this slide's a bit much. This is an example of what they were doing with their denitrification. So this was their plan. I pulled this directly off their website. And in a gist, I don't really want to read all this, but in a gist, they have bacteria, and this is the system that it's following. So your environment, at least the environment they were working with, was highly contaminated with nitrate, which is NO3 minus. And so they wanted bacteria to first sense, is there sufficient nitrate in the environment above a certain threshold to start turning on the system. So that's the first thing. I think I pulled this from the website. I think this is actually a typo, though. I think that should be nitrate. So right here you have two repressors, NAR and NSRR, and these repressors repress this promoter, and this promoter transcribes SERA. SERA codes for serine. Serine, yes, which is an amino acid. It's just what bacteria use for in order to live and grow. So it's very, very, it's common in at least this type of bacteria, and they can't live without it. So once these bacteria are added to the environment, the NO, so the nitric oxide present in the environment along with the nitrate, are going to de-repress these repressors. And because these repressors got de-repressed, this promoter is going to enact, which in turn transcribes serine. Seronin, the amino acid starts coding, starts being produced within the cell, and this in turn causes the cell to become healthy and start growing and duplicating. Once cells reach sufficient health, then NAPDABC, which is just an operon, starts coding for an enzyme that degrades nitrate into nitrite. And you can see that here. This is the promoter that they used for that. So as it starts degrading it, the nitrate ions are going to begin to decrease, which in turn tells the system to start turning off, because there's that threshold, right? So we don't want to completely deplete the nitrate from the environment, because that's also bad. We just want to get it to a like a sort of medium, to where it's sufficient for native plants to actually grow, but not poisonous to the aquatic environment. So once it reaches that medium point, this system is going to tell PAR to these repressors to enact again. It's going to shut off production of seronin, which in turn causes the cell to begin to pretty much die. It starts weakening, and then after it completely runs out of seronin, it can't do its natural processes, and therefore it dies. And so that's sort of its kill switch, right? So they programmed a kill switch into their nitrate degrading system, which is something we have to take into account when we're doing these sort of things. So if we're trying to bioremediate, most of the time bioremediation is just fixing something that's in too high concentration, not necessarily get rid of it entirely. So we have to keep that in mind. And of note here, after the cell dies, of course, it stops producing the nitrate degrading enzyme, and also the nitrate atoms are no longer reduced, so they're no longer degraded. This was the more complicated example, though. I don't expect you to understand this flow chart. In fact, I don't even understand 3/4 of it, or 1/4 of it. I understand 3/4 of it. So just keep in mind that we have to set a limiter of how much we can biodegrade. Next, I wanted to include a Colorado team. This is Colorado School of Mines, and they had a different project, at least different than the ones the other projects I saw in 2019. And this one was a cadmium sensing and also reducing project. Cadmium is a heavy metal. It is poisonous to humans and aquatic life. And so what they wanted to do is, first they wanted to find if you had cadmium in their water or aqueous solution. And they also wanted to remove that. And so this is an example of a more simpler project. So in the sensing step, this is your biobrick. This is your promoter. And this promoter specifically is highly adaptive to cadmium. So if cadmium is present in the environment, it will turn this promoter on, and in turn start transcribing GFP. So you can get a response, a green fluorescence protein response, and therefore measure that intensity in order to estimate, I guess, how much cadmium you have in your solution. This is very similar to what we're doing in our project this year. Most teams use GFP in this way. So if we need help, or if we need some sort of system in order to figure out how to get this working, we could always follow other teams' example. And this is the more complicated side of this project. This is the binding step. As you can see, you have your biobrick prefixes. Those carry over, of course. There's terminators. And then this is a lac promoter. So this is just a constitutive promoter, so it's always on. They did not finish their project entirely. Their goal was to eventually take this promoter and put it in this part, as you can see here. But for the sake of overlapping systems, which would have complicated their project, they just did these two things individually. So this is a constitutive promoter. It's always on. So that means it always transcribes these two proteins. And what those proteins are proteins that form in the periplasm of the cell, so in between the cell membrane. And it allows the cell, the bacterial cell, to attach to the cadmium and thereby reduce its motility, so it doesn't go through the environment as fast, and also provide the ability to skim the bacteria from the environment and thereby remove the cadmium. So overall, I don't think these projects are too complicated. I think it's pretty straightforward. You have your promoter and your protein coding sequences. It's definitely a lot simpler than this pathway. Colorado is also a team. They typically work within their own state. So Colorado has about 2 to 3 different iGEM teams. So they typically work with those teams, but we could even reach out to them if we wanted help or if we wanted guidance or even just to collaborate with them. I also included a few other teams. This is for your guys' viewing pleasure. So you can click on these. I will include the link, of course, in the video description. So I included a bunch of different types. I pulled these from the 2019 submission sheet. The first one is the PET microplastics degradation project. Yale did that. I actually watched that project during iGEM 2019 Jamboree. So a lot of teams actually did this PET degradation project because it's very, very popular because these microplastics are so prevalent in our environment. Then I also watched this one, Cornell. They did algal blooms. So algal blooms is what happens when you have tons of nitrogen fertilizer going into your lakes or your rivers. These algae, they eat that bacteria or that nitrogen like crazy. So what happens is they grow rapidly in population and they start blooming, which in turn produces, I think it's myotoxin. It's some sort of toxin that they produce that contaminates water, makes it poisonous for aquatic life and us. There's also simulating coral reef algae. So in the recent years, coral reefs were losing their color, and this was because the algae that were living in the coral reefs were dying due to increasing temperatures and like salinity and stuff like that in the oceans. So what they did is they created algae that was more resistant to these sudden changes, and Georgia State did that. I'm not going to go through all these, but yeah, methane reduction, polystyrene conversion. So they wanted to turn styrofoam into compounds. I think it was the monomers, break it down into monomers so they could work with it much better. Also acid and rhizobacteria. Rhizobacteria are the bacteria that live within plant roots, so they help fix nitrogen for the plants. And this was to make those rhizobacteria more effective at fixing nitrogen for the plants. And also breaking down pests and herbicides.